today we have Vaitea and Armand. Vaitea will present his work on self-reproducing ribozymes, and I'll give the floor to him now.
Okay, thank you very much for having me today. So I will be presenting a project where we design catalytic RNAs that are able to self-reproduce, and this is using probabilistic generative models. So this is, as you may expect, related to the context of the origin of life and one of his scenarios, which is the one from the RNA world hypothesis. So the idea of the scenario is that life has emerged from RNA because this molecule is able to carry genetic information, but also perform catalysis. However, in this story, you need kind of special type of character, which are RNAs that can produce copies of themselves catalytically. So that's where you have an autocatalytic cycle in this case. And those RNAs are called RNA-based self-reproducers. And typically, what we are interested in this project is to find those type of RNAs, right? Up to date, experimentally, there are two really known systems that are able to perform this type of self-reproduction. The first one on the right is the Joyce system. But in this study, we are focusing more in the system represented in the left, which is the group 1 intron from the group 1 intron RNA from the Azoarchus bacteria. So group 1 intron RNA molecules, they perform in the cell a kind of regulation from their self-splicing activity. So what you see on the figure are RNA species that are varying by how they are fragmented. So initially, what you have in the reaction is a collection of 4 fragments of the RNA molecule that are self-assembling and then connected by ligation, such that in the end of the cycle we have a fully connected copy of the RNA molecule, so the group 1 intron from the Azoarchus bacteria. So the question that we are trying to answer in this project is whether this type of self-reproduction RNA-based is something exceptional. So which means that, well, this whole scenario will be a bit unlikely that you would have like RNA-based life emergence, or whether those RNA self-reproduction molecules are quite banal, which means that you can easily find them in the sequence space of RNAs. So out of like a random distribution or random mixture of RNAs. So to assess this, we try to sample this RNA sequence space. However, before digging into what we actually did, I'd like to show 2 works that have been done earlier. So the first one in the left is an experimental work where they screen a combinatorial library of RNAs for a function of chelation. And essentially what they showed is that, well, for a given function in the sequence space of RNAs, those molecules are somewhat organized into islands of functional sequences that are connected by really small bridges of functional sequences. So this means that if you want to find new or really different type of active sequences, you have to cross those small bridges, which makes the whole exploration quite difficult. The work on the right shows a kind of an application of this idea where they combine machine learning with experiments to navigate the sequence space for given functions, for a given function. However, this is this has been shown to be quite challenging to find really different molecules and with quite a lot of differences in terms of mutations. So in the end, we know that, well, exploring the sequence space is quite difficult. So our way to handle this was to first design multiple generative models for RNA sequences, and to sample sequences out of those models, which are probabilistic models on the space of sequences that will test by experimental screening with a catalytic assay. So that's the whole strategy. So to build our model, we first use evolutionary data, evolutionary information. So evolutionary information in this case means that we gather from a database natural sequences that performs the same catalytic function. So in vivo, this function is the self-splicing. So we were able to gather for Azoarchus sequence about 800 sequences from the AirFarm database. So in the left, you can see a kind of a phylogenetic tree that shows more or less how the data is structured. On the right side, you see an analysis of the multiple sequence alignment we obtained from this. So multiple sequence alignment is a data structure where you can see evolutionary events like mutations and insertions and deletions. And therefore, what you see here on the top are the propensity of each nucleotide in terms of mutation in the natural counterparts of Azoarchus in our ensemble of sequences. So throughout all the 200 nucleotide position of the sequence. However, the most important information that you can see here is in the bottom, you have blue bars that represent the fraction of gaps for each of those positions. The gaps in the context of multiple sequence alignment represent insertions or deletions. And typically what it means is that we have a huge fraction of our data that is composed of insertion, which is typically missing information. So we need a model that can handle information that is missing in the initial data. To model this evolutionary data, then we choose to use a model that is called direct coupling analysis, which has been borrowed from statistical physics. And the core idea of this model is instead of using the whole sequence, we'll use a single and pairwise statistical signature. So this model has already been quite successful at designing enzymes in 2020. So the model goes as follows. So you see here multiple sequence alignment. So you have many sequences across different species, and you can see gaps where you have insertions, deletions, and mutations. So the model first takes all the positions and derive a parameter that describes the propensity of having a nucleotide at a given position. So that's a so-called field. And then you have coupling parameters that describe the propensity or the strength of having two nucleotides at two positions. So then using those parameters, we can derive a probability for every single sequence that has a Boltzmann distribution form where you have the couplings and the field that forms the Hamiltonian of the system. And to parameterize this model, we use the so-called Boltzmann learning, which essentially is like performing many Monte Carlo sampling to fit the distribution of natural sequences with this functional form. So once we have this model and trained on the 800 sequences we gathered from natural databases, we first cross-validated the model on an independent dataset that has been produced earlier by one of the collaborators. And essentially, this dataset is composed of sequences divided into 4 different selection pressures. So one piece of those sequences was highly selected with a very low concentration of ions in this case. So the concentration of ions is really important for maintaining the 3D structure of the RNA, but also for the reaction because it's one of the cofactors of the reaction. So what we did, we applied the model on all the sequences and to extract it's to compute a score, a DCA score. So what you see here is a distribution of DCA scores across the sequences for all the different selection pressures conditions. And what we can see is that we are able with this score to recover the trend that we would expect. So you have in the red line the wild type score, so we grounded at 0, and the most the sequences that have undergone like a heavy selection pressure have a better score, so are closer to the 0, and the sequences that went through less selection pressure. Then we perform a second cross-validation by using the parameters of the DCA model to infer contacts in the 3D structure of the RNA. So we know the 3D structure, which has been characterized by X-ray experiments. So what you're seeing here is the contact map that is known, that has been extracted from the structure in the bottom triangle, whereas you can see on the top triangle the prediction or the inference from the DCA model parameters. So out of these parameter, we can use the coupling terms to infer contacts, and that's what you see in green. And indeed, with this model, we were able to recover most of the secondary structure interaction, which are base pair interactions, stacking interactions, but also more subtle type of interaction where you have kissing loops and or tertiary interactions. So evolutionary information is not the only type of source of information we can use to sample sequences. The other part of the signal comes from its actual molecular structure. And there are models that do not use evolutionary information to predict the structure. So we have used one of them to predict the secondary structure. So this model works with parameters that have been obtained from melting optical melting temperature and used with a dynamic programming algorithm. And this combination allows you to predict the probability of two positions to be paired in the structural ensemble of a single sequence. So what you see in the heat map here are in the lower triangle the actual two contacts that we extracted from the known secondary structure of the wild type sequence. And in the upper triangle, you see the prediction of probabilities of position to be paired in the 3D structure ensemble for a given sequence. So the first thing is that we see that the prediction is quite accurately recovering most of the secondary structures. However, in the natural structure of Azoacris, there is a structural element that is called pseudonode, and that is really challenging computationally to take into account. So instead of using an algorithm that takes them into account explicitly, we use another one that is much faster, order of magnitude faster, but that do not take them into account. But we use the residual probabilities to indirectly account for the pseudonode. And the idea is to use this probability map to assess whether a sequence is likely to fold into the wild type structure. So to do that, we compute the Euclidean distance between the prediction map and the actual true contact maps.
Excuse me, I don't know
yep.
I don't know what a pseudonode is.
Oh, it's a base pairing interaction, but
yes.
So how to say, it's not different from the other type of interaction. It's the algorithm works with a dynamic programming scheme, so you need to be able to separate the interaction in the form of a tree. So the pseudonode somehow form a cycle in this architecture, and therefore you cannot easily decompose this calculation into a tree, and therefore you have to use another type of algorithm to compute the interactions. the
Can you
structure.
draw a pseudo knot, what it looks like in the air with your finger?
So
We
you
see
have
the
to,
camera. I mean,
that will be a bit difficult by hand, but I mean, I will show a structure in the end where you will see the
oh,
pseudo knot.
okay, okay. Sorry for interrupting. Okay.
No problem. Okay, so then if I come back to the structure-based score, we performed the same benchmark. So we computed this score based on the structure on the 4 level of selection pressure dataset, and you see the histogram that we derive on the right. And typically we observe the same thing, the same consistent result. However, a bit less striking than with the DCA. But we see that the sequences that are highly selected are closer to the wild type sequence score than the sequences that are not selected. Okay, now we have the potential to how to navigate the sequence space. So now I'll just briefly summarize basically how we do sample sequences. So initially we had those natural sequences with gaps and insertions, as I mentioned earlier, that we cropped to have data only modeling the wild type sequence. So we crop all the positions that are not involved in the wild type sequence. And that's the data we use to train the DCA model. Then on the other side, we have the thermodynamic prediction of the secondary structure of RNAs that allows us to measure how well a structure will fold into the wild type structure that is known to be compatible with the catalytic function. And there we can use those two scores as potential to guide the exploration. And for the exploration, we use Markov chain Monte Carlo samplings, where essentially we'll reweight the contribution of those two directions, either evolutionary based or either somehow physics based direction by reweighting their contribution. So now we are able to sample sequences, but how do we test them? So in the lab, we have developed a catalytic assay first for the self-splicing reaction. So as I mentioned earlier, self-splicing reaction is the natural function of group 1 entries. And we'll use first this reaction as a proxy reaction for the self-reproduction that we'll test later. The reason is that for the self-splicing reaction, we are able now to perform a high-throughput catalytic assay where we can test thousands of sequences for this function that we thought is a necessary condition for having self-reproduction. So what we do, we generate computationally sequences that we synthesize with the exon of the wild type sequence attached in the 3' of the 3' of the design sequences. So here you see sequences, and in the end we see a green piece of oligonucleotide that is the exon. Then we incubate those design sequences with a substrate that is also an oligonucleotide with a sequence that we can recognize. And then the idea is that active sequences, they will be able to remove the exon and ligate the substrate. And then in the end, we'll perform a PCR selection, which essentially will amplify the sequences that have the substrate in the end. And then we'll identify all the active sequences by deep sequencing. And to measure the activity of all the designs, we compute the frequencies after and before the selection, and compute the log ratio of the two frequencies to compute their catalytic activity for the self-splicing. And later on, I show how we validate this self-splicing as a as a proxy reaction for the self-splicing, for the self-production. Sorry. So using the models, we sampled about 40,000 RNA sequences that we tested experimentally with first the self-splicing high-throughput catalytic assay. So first, I'll show you a comparison between sequences that have been designed fully randomly. So it means that we take position in the sequence randomly out of the uniform distribution, and similarly, we mutate them totally randomly into the 3 remaining nucleotides. And that's how we perform random mutations. So the figure you see on the right shows the active fraction. So how many sequences were found active in the self-splicing at various mutation distances. So number of mutation from the wild-type sequence. So you can see first that using fully random mutations, you kind of quickly lose the catalytic activity. So after about 15 mutations, most of the sequences we designed above that number of mutations were found totally inactive. When we use the structure-based score that I described earlier, we found out that actually we're not able to sample sequences active much further away from the wild type, which somehow is quite expected because this model of secondary structure does not carry direct information about the catalysis. However, if you use the evolutionary model that is inside the DCA model, then we are able to detect active sequences up to 55 mutation away from the wild type. So what it says essentially is that, well, even though you don't have like an explicit information of the catalysis, the sequences that have been sampled, naturally they are conditioned by having this function, right? And we can see that by comparing the 2 scores with the activity that we measured by sequencing, and we see that sequences with a low DCA score, which is a good score, have more active sequences than the ones that are generated with a higher DCA score. However, this relation, you don't find it with the structure-based score. Then we tested also this to modify the sampling temperature. This is a kind of a fake evolutionary temperature that modulates the diversity of sequences that you can generate. And if you reduce it, what we saw is that you can generate sequences much further away from the wild-type sequence, but at the cost of diversity. So essentially, what happens is that you have more or less the same sequence that is that have like a few mutation. So it's essentially always the same sequence. So this success rate is somehow inflated by the loss of diversity of sequences. Then we tested another hypothesis that is related to the structure of the model. So the DCA model is a coupling, so pairwise model. However, it is thought that you may need higher order interactions to model catalysis, or at least RNA molecules. So we tested a neural network-based model that is called variational autoencoder. However, this approach yielded the same result than the DCA at temperature 1. So it means that a secondary pairwise interaction in this context is sufficient to model these data. So these higher order interactions hypothesis comes from the idea that, well, for protein enzymes, you need specific type of tertiary interaction to reduce the activation energy barrier to facilitate the reaction through the transition state. So to test this hypothesis, we have frozen the position involved in 3D structures interactions, so they are not allowed to mutate, and the rest of the position were allowed to mutate using the secondary structure score. And we can see that we can generate now, with the same amount of mutation, more active sequences than using only the secondary structure score. However, we also tested not using the secondary structure, but only performing compatible sequence sampling. So this means that at paired position, we will sample only canonical base pairs. And somehow we obtain the same result then with the structure score. And this aligns with the result that I showed earlier, that the second structure score we used here is not aligned with the catalysis in this function. And finally, we tried to combine those two directions, so evolutionary based and physically based potential with the Monte Carlo sampling by reweighting the two contributions. And using this, we were able to sample even further away from the wild type to like 65 mutations away from the Azoarchus sequence. So the question here is whether the sequences that we were able to sample are actually really different from the natural ones, or are they somehow similar? To test this, what we did was to take the DCA alone, the DCA sequences generated, and to train a PCA representation. So using those axes, we projected the sequences we generated with the DCA only. The active ones are shown in blue, and the gray one are the inactive generated sequences. On the other plot, you see the sequences generated by DCA with second structure. And somehow this shows that the two ensemble of sequences are spanning the same space. Then we did the opposite. We take the DCA plus structure sequences and train a PCA representation. And what we can see is that in this group of, in this projection, we have a cluster of sequences that is not sampled if you use only the DCA scores, right? And actually those sequences, they are sampled because they have a structure score that is much more favorable. So they are more stable to fold into the wild type structure. So those sequences, they actually have a quite different statistical signature from the evolutionary one. So where those mutations are actually performed. So as you asked earlier, the pseudonode is this section here. So it's called the P3 loop. And you see that they overlap the path of another secondary structure element. That's what that's the so-called pseudonode. And this structure here is divided into two regions. So you have a yellow region that is called the scaffold, and its role is to stabilize the geometry of the molecule. And the gray part is called the catalytic core, which holds the catalytic nucleotides. And so what the color scheme here indicates in red that the position has been mutated a lot in our designs, whereas positions in green indicates nucleotides that were not mutated or not heavily mutated in our designs. And
I apologize for nagging you again. A pseudonode refers to non-planarity, or to the
exactly
appearance of cycles, which is different things.
No, it's the planarity.
Non-planarity, thank you.
Yeah. And well, this shows that typically in the catalytic core that is here in the P3 loop, we were not able to introduce much mutations. So the catalytic core is actually quite difficult to mutate with the model we used. However, we are able to vary quite a lot the other parts of the molecule, like the scaffold, for example, that holds this structural role. So now that we have, we were able to screen for the self-splicing, the question is whether this self-splicing is actually a good proxy for the self-reproduction that we are interested in. To test this, we took several sequences that were active in the self-splicing, measured by the sequencing score that we have. And then we perform a self-reproduction assay, which essentially is cutting sequences into fragments and incubating them to see whether the molecule is able to self-assemble and then perform ligation to create fully covalent copies of the initial molecule. So we can do it in 2 fragments or 4 fragments. We take a couple of few sequences, and then indeed, after the experiment, we found out that, well, the sequences that have a high self-splicing score were actually also active for the self-reproduction. We tested also the 4-fragment self-reproduction that have been originally studied in for Azoarcus systems, and we found a couple of sequences that also are able to perform this 4-fragment self-reproduction throughout this study. So to come now to answering or trying to give an answer to the question whether this function is rare or not in the sequence space, we measure the support size of the DCA model, so the entropy of the model. That gives us how many different sequences we can sample out of this model. And then we corrected this number by the experimental result we have so far, and we obtain the result we have is that we can generate potentially 10 to the 39 self-reproducers from this DCA model in the sequence space that is somehow delineated by the size of the Azoarcus system. And we can do this calculation up to about 65 mutation because that's the farthest away we were able to design an active sequence from the wild-type sequence. So that's the last part of my presentation. So I'd like, before the end, to thank the people that worked on this project. So all the experimental part has been done by Camille. Francesco, that worked with Martin White in Sorbonne University, worked on the one part of the DCA model that was used to compute the support size and generate some of the sequences. And then I worked with Matteo Smerlak in the MPI to generate sequences along with Philippe Enville. And I'd like to thank you for your attention.